Yes, sir. Yes.
Okay. So good afternoon, everybody. For this particular talk, we have Dr. Ashwani Sharma. He is currently CEO of Inside BioSolutions, France, and MD Moldog Biotech Private Limited, India. Dr. Ashwani Sharma has more than 15 years of working experience in the field of computational chemistry and biology for solving biotech industry problems. After PhD in bioinformatics, he worked with European companies for safety assessment of pharma and cosmetic product using molecular modeling and machine learning techniques, along with the commercialization of the technology. As a member of the board and president of the scientific committee of the French Society, Group of Metabolism and Pharmacokinetics, GMP, and European Society for Alternative Animal Testing, USAT, Dr. Ashwani provides support for using in vitro and in silico model for safety assessment of pharma and cosmetic products. So welcome, Dr. Ashwani. Now you can start your talk.
Yeah, thank you very much, Dr. Sumer, and then plus also the organizing committee. It's very grateful for such kind of invitation. And then I can say that I'm associated with this institution with some memories since my almost 7 and 8 years. But now with Dr. Sumer Singh, who were our colleague before, it's very privileged for this invitation. So first of all, thank you very much to you and all the organizing committee. So I will share my slide. So my camera is working from my company profile, and then the another one, so I have it too. So that's why I'm now sharing slide with the Ashwani Sharma profile.
Okay, sir, we cannot see your picture, your video. Can you start your video?
Yeah, because from the slide which I'm sharing, so in this the camera is it's because of security is hidden, but I shared with my company profile, so you can see where Inside BioSolution is there, so you can see me to interact.
Okay, fine, no problem.
Okay, so can you see my slides now?
Yes, we can see. We can see your slides.
Yeah, so thank you very much for such a brief information about the topic which I'm going to speak. So as now I'm CEO of Inside BioSolutions France. So it's a company located in the western part of France. And now it's 2.5 years successfully we are working to promote non-animal testing. So what exactly the non-animal testing and why this topic is major concern related to especially in Europe, in also in America, and now well versed into several Asiatic countries. So first of all, the title of my topic, so it's a global topic, but now I'm talking about machine learning and artificial intelligence method and how these methods are implicated to produce and analyze the drug toxicity. When you are talking about the drugs, so there are plenty of different toxicity you have to evaluate at the pre-screening stage. So when you will talk about the drug toxicity and it's an end effect of the drug, but at the pre-screening stage, you have to evaluate all those drug toxicity so that the drug which you are starting from, let's say 10,000 molecules, maybe million of molecules, and the stage which is lead identification, it has to pass to 3 pre-clinical screening. And during this pre-clinical screening, when you have plenty of drugs or chemicals are come up with the toxicity, then it has to filter and hardly 100 drugs are going at the stage of like a first phase of animal testing, then after that clinical trial. So pre-clinical screening is one of the most important steps to understand the safety of drug. And here, plenty of different methods are applied. And one of them now extensively used machine learning models. And I'm here in this title and in this presentation talking about how these machine learning based methods are involved in analyzing genotoxicity. So genetic material influenced by the drug. So that toxicity is called genotoxicity. So we developed this prediction model by using these methods. Now to have a little bit of information, so that's our beautiful campus where we are located in the western part of France, and it's a biotech park. We have a plenty of companies coming from different pharmaceutically, plus cosmetic industries. So we are merging together these two domains, pharma and cosmetics, because many of the chemicals, when we are working towards developing some pharma and also the cosmetic, they are sharing together these chemicals. So that's become very important to understand safety of those chemicals. So our company are located in this biotech park where we are connected with pharma and cosmetic, and to provide information by our innovative methods, which we integrated by silico and experiment, how your products are safe. In our team, we are core 8 people, and out of that, we have some other technician working to provide this opportunity. And what is the mission and goal, and why it became very important from Indian prospect? That's the reason I'm going to explain here. Like, the mission and goal is to develop alternative methods. So I'm more non-animal tester and promotion of the non-animal testing methods. So that's why personally we developed the goal to develop some computational method, integration with some experiment, and these two techniques comes together, we can analyze major part of the safety for either cosmetic ingredient or maybe for pharma products, and that is the goal we developed this company. Now, as here we are talking about pharma, so drug toxicity. So what exactly drug toxicity? So drug toxicity usually influence when we have talking, okay, this drug is allergy to us. This drug is creating that side effect. So usually we are the consumer, we are the user, and this information will go back and talk about, sorry, your drug is very toxic and you have to withdraw these drugs from the market. If people like, it's very good to interact with the excellent scientist and professor, because without that, I can purely say industry doesn't work. So when you will go with the trends from industry, we always get prospects, sorry, your drug is toxic because of that reason, because of maybe liver toxicity, because of kidney toxicity, because of neural toxicity, you have several body blood-brain barrier if it is developed. So many prospects to reject the drug. And why this is happening? First, the tools were not available. 2, when we perform animal testing, it's too expensive, it's too troublesome, and now we have so many ethical aspects. And because of this, what we call drug toxicity, many of the side effects are ignored at the preclinical scale. So drug toxicity is very important to test where a particular chemical drug is damaging your particular organism. Why I'm talking about organism? Because when we are testing these drugs on animals, which is still going on at some stage, if you will talk about testing on animals, extrapolation of that data to human, it's become a little bit very difficult. So because of this, the drug toxicity are not tested perfectly, precisely, and when we used as a consumer, as a human, we are getting several side effects. And these toxic effects of the drug are obviously dose dependent, and that damage either the whole system or damage a specific organ. So that's the meaning of drug toxicity. And why this drug toxicity used to happen, as I said, drug toxicity is a global term. When you take the drug, you start from the mouth, and then it goes to your particular alimentary system. And after the alimentary system, it has to absorb through the membrane, and after that, it has to interact with your particular target. After interaction with particular target, it has to eliminate. So the drug toxicity used to come under one term, which we call in pharmaceutical industry, ADME, absorption, distribution, metabolism, and elimination. And each step is very important to analyze whether your drug is toxic or not. And that's why when you talk about the drug toxicity, the drug going inside your mouth, it's a parental compound, it's a complete formulation. And when this complete formulation is going inside your body, either sometimes the substances are metabolized. So there are certain organs in the body which are performing metabolism. And after this metabolism, the drugs are interacting with your specific receptor. And that's the stage we have to evaluate also. So there are two kinds of receptor. Either they are intended receptor, you have correct receptors, but these current target receptor are having problem related to the chemical entity of the drug, high dose of the drug, or activation or inhibition. But as you have drug metabolites, then it can interact with your unintended receptor, and then they activate incurrent receptor. And if it is not, then you have an unintended receptor. And based on this, you go for the correct receptor, but incorrect site. So drug toxicity, as we said, is a global term, but the scenario to test every aspect of the drug toxicity. And why it was happening? First, limited resources, long time, animal testing is not extrapolated to the human-based behavior, and plus also many times we have a low budget, we are ignoring to testing these kind of toxicity. So it's become very important to perform new and new development of assays, other methods for drug toxicity assessment. Now, one of the toxicity, when you are talking about all, in case of pharma, it's obligation and important to test genotoxicity. And what exactly the genotoxicity? The terms itself in definition. You have one particular chemical entity which is in the formulation. When you take inside your body, it will directly interact with your DNA also. So any drug which is creating destructive effect on the genetic material, that called genotoxicity. So they can influence by several behavior on the DNA. So as I said, the drug, when it is going, every drug, every chemical, when you are taking inside the body, in the same way, partial dermocosmetic products, when you are taking wound healing, all those are coming into the category of pharma by certain territories. Like in India, FDA regulate, so we call Indian FDA system, Central Drug Regulatory Authority, they regulate drug and cosmetic at the same level. So when we are talking about the drug, it has to go ADME pathway, absorption. So you absorb the drug, distribution, it will distribute where exactly the target of this drug, then metabolism. Liver has a first site of metabolism, and then after that, it will metabolite into component and divide. And why you have to analyze the genotoxicity? After the metabolism, every drug is going to interact with your DNA. And then based on this, you have a two category of genotoxicity assessment. Primary genotoxicity. Many times we are performing research and we are talking about whether my drug is creating inhibition effect on the DNA replication enzyme, DNA repair, cell cycle control, so on, because we work extensively on oncology. So that kind of effect, it comes also into genotoxicity category. And we have direct DNA damage. Your drug is directly interacting with your DNA and then damaging the double-stranded DNA and also into RNA. So that kind of genotoxicity is coming into primary genotoxicity. Free radical formation is one of the hotspot criteria in pharmaceutical world and the cosmetic world, that whether your drug is creating free radicals damaging the DNA. And it's one of the high-tech time for pharmaceutical industry to calculate reactive oxygen species. But if you say, okay, now my drug is not in the primary genotoxicity pathway, maybe it is going towards secondary genotoxicity pathway. And that secondary genotoxicity pathway are like inflammation, you have immunological response, you have some receptor applications, and that can also evoked by reactive oxygen species. And this oxidation DNA damage again going to genotoxicity. So you can see when you analyze different aspects, we performed several research on DNA enzymes, oncology, everything is ending towards genotoxicity. And genotoxicity are measured all the mutation you are having, carcinogenicity, mutagenicity, several aspects. So that's why it becomes obligation. Any person who are performing drug screening, they have to analyze genotoxicity. I work with the regulatory agency here in EMA and also registration of product in FDA. Without this data, we don't recommend to submit any application for registration of product into market. And that's the important. Now, why the non-animal testing? Before, it was quite easier. You inject the drug into the animals, and then plus also you can analyze. But as we see that during school time, we also perform these animal testing in engineering time. Now, based on new norms, that these animal data are sometimes not relevant to have human data, or only animal testing is not sufficient. Cosmetic completely banned animal testing after 2014. And also in India, it's strongly implemented. You cannot launch any chemical molecules tested on the animal for cosmetic product. For pharma, now the legislation passed to use alternative method. And after 2025, you have to reduce 50% of animal testing for pharma product development. Otherwise, your drug will not be in the market. So based on this, European regulation implemented new methodology, and that is called New Approach Methodology. We call NAM. So any method, like you perform cell cultures from since long, that's come under the category of new approach methodology. You test the drug on cell cultures, you test the drug at the plant level, plant tissue culture, all are coming under the category of new approach methodology. So that's why the animal used to say, now you have to use flies, worms, human cells, and the computer. Recently, just yesterday, FDA passed the legislation. Computational method can be used and validated to perform any product coming into the market. So that's a very good news that FDA will recommend to use these methods. Now, why this genotoxicity? So now you can see legislation always control the assay, and these assay are always reported into agency, and you have to follow the procedure. Pharmaceutical industry are always attached with academic, and academic is very important. Private-public partnership is always very important to update these guidelines, because academic can perform better research as compared to industry. And when these guidelines are launched, so for genotoxicity, the specific guidelines are ICH-M7 guidelines. And ICH-M7 guidelines are talking about carcinogenicity, mutagenicity, any damage to the oncology. So suppose if you are developing drugs for oncology, so you have to evaluate all. And any manufacturing, so we have lots of generic products manufacturing, its obligation to perform and follow these guidelines for genotoxicity assessment. And the process you have to follow is just highlight a little bit. You have to precisely purify your product. There should be no impurities. And if you have impurities, the label should be very, very low. And you have to disclose you have impurities. And that's many times like some drugs are withdrawn into the market after used 10 years, 15 years, because yes, before method were not available. Now you have high LCM SMS to check all the purification. So like this, you can use those technology again and to understand whether your compound is very much purified or not, you have any contamination or not, like even single stage of pesticide impurity in water, it can create genotoxicity. So it's very important to report this into the application before registering your product into EMA or FDA, and that's important. And then once you have these products, impurities, you have to characterize whether your products are creating any DNA damage or not. So this is a global term when you are doing manufacturing. Then when you perform some assay, important assay are always updated in these guidelines. So for genotoxicity, every time the consortium known as OECD, it's in Europe, and they always release the guidelines, and these guidelines has to follow globally who are connected with OECD. India is the partner of OECD, so India also perform all the assay based on the OECD when any company providing support to industrial production, industrial assessment, industrial preclinical screening. Academic also participate if they follow OECD guidelines. There are certain requirements of laboratory, but they can assist into this. So when you want to analyze genotoxicity, there are very well-known 4 assays based on these guidelines. So first, you can have you can use mutant variant, and if your drug is reversing this mutation, so you have a positive sign of genotoxicity. Then you have, if there is a problem related to thymidine kinase, so selection of gene mutation can give you answer of genotoxicity. When you have a nuclear aberration and the chromosomal aberration, that's a positive sign for your drug that your drug will be a potential genotoxicity. So if you follow all the procedure, you are able to detect genotoxicity. And that's the reason the guidelines are always updated and accept, okay, if somebody follow, then it's perfectly fine for your product to get a good result. Now, based on different industry, we have to split the assay criteria. If you are cosmetic, you have to perform in vitro test. If you are chemical and bioseed, which are very strong genotoxicity component, you can use in vitro test, but at least perform one in vivo. Then if you have plant product, veterinary drug, they are strongly implementing the same procedure. And when you have a pharmaceuticals, you have a two option. First, you perform two assay, one in vivo test, or you perform one in vitro test and also perform in vivo assessment. So these are the specific criteria you have to perform and analyze for genotoxicity. Now, based on this, which assay? So I'm not going into detail here, but one of the consortium along with the OECD. So we work with PETA Europe and also now slowly start working in PETA India to help pharmaceutical industry, which assay you should use instead of animal testing. So slowly you will reduce, because now you have product in the market. After 2025, you have to withdraw the product from the market. So they are already aware about that, to use as much as assay-based method and computational method. And that's a good lucky part, like from 2017 onwards, the strategy implemented, you should use computational method also, integration with experiment. So now EUR ECWAM, who is controller of all over the world assays, they recommended in silico method should be integrated with in vitro or in vivo method to at least provide more and more predictability of the data, and plus more for this genotoxicity. And that's the lucky point from where we started a consortium, and to have a decision, we should develop a own innovative method for performing several toxicity. One of them is genotoxicity, which I will just explain in few slides. Now, if you talk about the computational method, so what was the computational method we exploited? So usually we use several computational methods since long in the pharmaceutical industry. Molecular modeling is highly exploited. So the first layer is the molecular modeling. We perform interaction analysis. So we check the chemical properties, we do molecular docking, molecular dynamics, and now highly throughput computational chemistry. Then after that, the first category, which was long time ago used in preclinical screening, was the QSAR, quantitative structure activity relationship, and the read across. So these two were quite exploited into pharmaceutical industry. The first one used in the lead optimization. That's why at the first time I use lead optimization, means out of 1,000, select some certain drugs. Then these drugs will go for QSAR analysis at preclinical screening. That was the two exploited since long time. Now, the new method aware from last 2-3 years to use prediction models by machine learning and artificial intelligence. So what these methods used to do, so they collect the data, they develop chemical properties and generate the model and perform the prediction. But all these three layers were separated. What is the point of Inside BioSolution? How we delight to develop innovative models? So there are certain assay or there are software which are performing QSAR based. So currently most of the methods are QSAR based, as I given that the software which was since beginning for QSAR based on the property matching. And when you will look about the QSAR, just a very small description. If you have a chemical, you know the compound is toxic, so you know why, because of chemical properties. When you have a new compound, you try to match with existing chemical compound who are toxic or non-toxic. Based on this matching percentage, you decide whether your compound is toxic or not. So very simple, means you check the chemical properties matching for new compound with existing compound, and based on the existing compound, you define your compound is toxic or not. And that's the QSAR, which used and not exploited. What IBS is doing now, we are integrating all the three layers. So here you will see, so where you are using integration of all the method, machine learning based algorithm using for molecular modeling, QSAR, read across, and developing own prediction model. And that's the power from us. So we develop all those prediction model. We collected lots of in vitro and in vivo data from clinics, FDA and EMA. After collection of all those data, we take the chemical properties also, and we combine these properties by the help of machine learning and develop our first pilot innovated model for genotoxicity. So that's become kind of very accurate soon. Now, why to develop these methods? Because if you have, let's say, 50 compound to test by pharmaceutical industry, because they have 1,000. If you want to test the compounds, I can give you just one simple criteria. One compound testing, currently the assay for genotoxicity is more than 15 lakh, just one compound. Now you can multiply by 50. You have to perform this assay triplet, so 3 times, and then further goes on in 6 concentration. So now you can see how much money people are spending to perform just one toxicity. Why this in silico method and why FDA, EMA are recommended? Because the data is not precise with the experiment. It's not so accurate for humans. So we have to use different alternative methods. So by in silico, it's very more rapid and reliable prediction, and it reduces the time and cost. And then after selection of compound, you can further validate selective compound by in vitro experiment. And currently in the pilot project, what we have did, we collected 442 compounds for which we have a clinical data also, and we collected the properties from PubChem and plus some Chem Spider. After collection and selection of those 442 compounds, we took care of very advanced descriptor. Out of 30, we found 8 are very important: molecular weight, log P, pK value, hydrogen bond, donor, acceptor. And in case of computer, it accepts only binary classification. Binary means 1 and 0. It cannot give you very answer in the beginning stage. So we classify binary classification. If after evaluation of the compound internally into the algorithm, if the compound value is coming 1 based on the property matching, the compound is genotoxic. If it is coming 0, the compound is non-genotoxic. So that's one of the criteria. Now, this is the description, like interaction between 2 categories, in vitro and in vivo. Physicochemical data, when you integrate together by the help of machine learning, which can handle as many as descriptor, then you have to select the best descriptor, then it will give you genotoxicity or non-genotoxicity. Now, this is the flow. We took 442 FDA-approved drug. We collected clinical data in vitro, and in vivo means from patient data, from FDA, from EMA. So we collected several descriptors. After that, we collected all the compound physicochemical properties. Always remember, in these kind of machine learning, if you select 10 compounds, 50 compounds, if you select certain descriptor, you need all. You cannot have blank, so that's the problem. So the most important, the data which you are putting into the machine learning. Then after that, we split the data into 2 categories. One, to develop training model. Training model means you select certain compound definition, genotoxic, non-genotoxic, or medium. Then we collected 330 compound approx to use as a training model. Then we wanted to see our model is good or not. We took like 111 compound for testing. We know there are genotoxicity value, but we consider let's our model to predict. And based on this, we used several machine learning algorithm, not to be biased that which one is useful or not. So we collected several machine learning algorithm to understand what is the value of genotoxicity and non-genotoxicity is coming. Here, the results. When you do prediction model, so we successfully derive and separated the compound which are genotoxic, which are non-genotoxic. So we can develop these criteria in a graphical representation, like the compound in blue are non-genotoxic and the compound in orange are genotoxic. And we separated based on this property. When the compound is genotoxic, what is the distribution related to their properties? So here we found the compound which are coming genotoxic always into the market, they are slightly lighter in weight as compared to non-genotoxic compound. And the compound which are having smaller rotable bond counts are considered as a non-genotoxic, in the same manner for hydrogen bond system. So now by this algorithm and our prediction, we can clearly by graphical representation can see the compound is genotoxic or non-genotoxic. Then when we are thinking, okay, now we need to implement efficacy. When you do this prediction, we call this is prediction, so safety. Now you have to do efficacy. So for that, we develop the accuracy chart, and now we have to update more and more. Till now, the model which they developed, so many times they have around 75 to 80%. Now with our model, so we found 3 methods are closer to 80+%. The prediction of accuracy, specificity, and sensitivity. And when we perform false and true positive rate calculation, we found these methods are giving true positive, means accurately result around 90% value. So means, but we have to now improve more and more. We collected more data for around 900 compounds to improve the efficiency at least 90% to 95%. So in this, we are very much in a good direction that we collected, we predict the model, our algorithms are trained. Now we have to feed more and more data coming from the experiment. So that's why we said private-public partnership is very important. The data integration is very important to work together. Now, once you perform this analysis, the most important was to efficacy in pharmaceutical or in cosmetic world, how they interact, how they behave. So in this situation, we use the traditional method. We collected those compounds which we have collected before. Then the most important in efficacy, how they interact with the DNA. Here, I'm giving very basic information by using molecular docking. Now we are performing with computational chemistry because it's a heavy machine learning task. Machine task. So in case of this, first what we did, the compound which we have selected before here. So we selected those compounds. We found which compound are genotoxic and non-genotoxic. So we selected top compounds. And then after those drugs, we perform molecular docking with DNA. If people who are not aware about molecular docking is to identify interaction between your macromolecules and your compound by computational method. Each interaction will give you binding energy, and lower is the binding energy, higher is the affinity. We assume the compound which are genotoxic is strongly bind with the DNA, and that's why they are blocking or creating problem for the DNA. Now, for that, we created currently 15 models. I'm here giving you 5 models because we don't know which genes or sequences are inside the body and which is the most target. And that's the first time we are performing in this kind of task. So we selected and giving the result here currently for 5 DNA models. So we randomly created based on the percentage of availability of DNA, and we collected this data from liver. What are the genes available in liver and how much gene sequence variation? So we found these 5 kind of sequences are available 80% in the liver. And once we have this data, we found the compound which are genotoxic. So you have several experimental genotoxicity data, like a chromosomal aberration, so which contain many activity inside that, or non-genotoxic, like a control. We found the compound which are genotoxic, they are highly interacting with the DNA with the low binding energy. So these are the drugs coming from FDA, and the compound which are non-genotoxic, they have a little bit less interaction as compared to the genotoxic drug. So that's we found that efficacy prediction became very important. Now we are doing this for all. We are collected total approximately 1,200 compounds to perform first collection of clinical data, integration in our machine learning model, performing molecular docking, further on computational chemistry analysis. Why? The one important point here, genotoxic currently we produce non-covalent binding. This is one criteria of interaction. Then you have an intercalation, and third criteria is covalent.
Sorry to interrupt in between, sir. So just wanted to know, for non-genotoxic and for genotoxic, the difference is not much. Like it is, if it is like for 6.9 for them, but here it is -8. So the difference is not much. How will you say this is non-genotoxic and this is a significant interaction? This is not significant.
Yeah, so that's why that's why just I immediately inform that this is a first by the molecular docking, and in molecular docking is the first stage of simulation, and now we improve this system by the free energy simulation by molecular dynamics, and from molecular dynamics we start getting very much higher difference, because in molecular docking first you can get only pre-click. Pre-click means which pose you can collect to put into the molecular dynamics. And then after that, the last and final improvement by computational chemistry method. And by computational chemistry, we found like here you have a -6, and this pose we submitted into computational chemistry simulation. And because this data is hidden, it's industrial data. And we found the difference of at least between genotoxic and non-genotoxic compound +5, means you can say the difference of 5 factor. So that's the big difference we found. We were able to detect, even because in this we cannot perform intercalation. Molecular docking cannot perform intercalation, but molecular dynamics, for example, into the compound of acetylbutolona, here we found it's intercalated also. So it's a non-covalent binding by and the spiral binding, but it went into the intercalation. And this intercalation make more difference between non-genotoxic and genotoxic drug. So that's why I was explaining here also, like mode of binding is very important. Every method has limitation to perform such kind of analysis like a in vitro method. And now the data is improving by computational chemistry, which are going to improve within 2 months. We are integrating in the final model, and that model will come for validation with in vitro experiment people. So in a conclusion, what we are saying that alternative methods are really important to implement to give as much as support for understanding the drug toxicity, like here we are doing for genotoxicity. This is one. What we are talking, and it's globally written into several guidelines, when the drug is going into genotoxicity, because before that people are talking if there is a carcinogenicity, it's genotoxicity. But then by the help of several research, several assays are added on to support. If you consider drug is genotoxic, you should analyze mutagenicity, you should analyze chromosomal aberration, you should want to analyze free radical. So all those data for one single drug became very important. Then the computational method add-on, you start working on QSAR, but they found chemical properties is not sufficient. Then the molecular docking came here, so where we are working interaction analysis. Then molecular dynamics came because non-covalent interaction is one part, but then intercalation. Then there is another category came. When you have a compound, it's many times not intercalation or like a binding, but now group binding, but also came exchanging electron, and the compound is interacting with the DNA and breaking the DNA, and that's not intercalation or group binding. And now the latest technology of computational chemistry can perform. So it's an involvement as much as support to the experiment to predict a alert before going for experiment, and to consume, like to reduce the time and cost for large number of drugs to filter the best drug. Now that's why in the future we are performing more molecular modeling analysis, how many descriptors we can collect from the experimental data. Now another one, which is category to calculate drug metabolites. Currently the drugs is available. Now the metabolites we have to create. So we created some information from drug metabolites, because sometimes drug is not genotoxic, but the metabolites are toxic. So that's why this will influence all in vitro experiment. This will influence all in silico experiment. So drug metabolites became very important. And also, as we said, free radical binding. So this is our future in next 2 to 3 months to integrate all those information to create very precise and good and powerful prediction model. So thank you very much. And then if any question and something, so you can contact on the email address. And then Dr. Sumay Singh Meena is also there. And our project is supported by the French Ministry and plus Emergis, and now also BPI funds. And now in next year, we are making Indo-French joint partnership for these kind of projects, where we are always working with academic and private-public partnership. So thank you very much. And then I'm happy to answer some question if you are here. Hope I'm on time.
Thank you, Dr. Ashwani, for wonderful talk. Now participant can ask questions, please.